Welcome to everyone. My name is A. Dinesh and I'm from 2nd MSc Biochemistry and my topic classification of liver functioning test LFT. Classification of liver functioning test. The liver functioning test used in the study of patient with liver and biliary tract disease can be classified according to the specific function of the liver involved. Test based on the abnormalities of bile pigment metabolism, test based on the liver part in the carbohydrates metabolism, test based on change in the plasma protein, test based on abnormalities of lipid, test based on detoxicating function of liver, excretion of injected substance by the liver, excretory function, formation of prothrombin by liver, test based on amino acid catabolism, test based on drug metabolism, determination of serum enzymes activities. Test based on abnormalities of bile pigment metabolism. The bile pigment metabolism, a group of pyrulic compounds that constitute the end product of the catabolism of immunoglobulin and possibly other related tetrapyrols. There are the three types of tests in the bile pigment metabolism. They are serum bilirubin and VD-Berg reaction, and urine bilirubin, and finally urine and fecal urobilinogen. Test based on liver part in carbohydrates metabolism. The liver play a central role maintaining the blood glucose level. Following consumption of food, excess glucose can be stored within the liver as a glycogen. This is stimulated by insulin release. Around 100 gram of glycogen stored in the liver, 300 grams stored in the skeletal muscles. There are two types of carbohydrates metabolism test. One galactose tolerance test and other one fructose tolerance test. And the next one, test based on change in the plasma protein. The plasma protein are the enzymes, while other have functions in the blood clotting and the immune system. Excluding immunoglobulin, all the major blood proteins are synthesized in the liver. The total protein test gives an approximate measure of all the plasma proteins. There are the four types of the test in the plasma proteins. They are estimation of total plasma protein, albumin and globulin, and the determination of A:G ratio, determination of plasma fibrinogen, and various flocculation tests, and finally amino acid in urine. And next one, test based on abnormalities of lipids. The liver synthesizes large quantity of cholesterol and phospholipids. A bulk of lipoprotein are synthesized in the liver. They are the two types for lipid abnormalities. They are determination of serum cholesterol and ester cholesterol and the ratio, and finally determination of fecal fat. And the next one, test based on detoxicating function of liver. A Kupffer cell of the liver remove the foreign bodies from the blood by phagocytosis. The liver cells can detoxicate drugs, hormones, and converted them into less toxic substance from the excretions. A test for detoxicating function of liver is hippuric acid synthesis test. And the next test is excretion of injected substance by the liver, excretory function. The certain exogenous dyes like BSP, bromsulfathione, and the rose bengal dyes are exclusively excreted through the liver cells. And next one, formation of prothrombin by liver. A prothrombin is a protein made by the liver. A prothrombin helps the blood to clot. There are the two types of the test. They are determination of prothrombin time and determination of prothrombin index. And the next one, test based on amino acid catabolism. The catabolism of amino acid, except those with branched chains, start in the liver. The amine group is separated and incorporated into the urea. The carbon skeletons can be oxidized to CO2 and H2O or used for gluconeogenesis and ketogenesis. Liver is very efficient in the removal of ammonia. The test for amino acid catabolisms, they are determination of blood NH3 and determination of glutamine in CS fluid. It is an indirect liver function test. And the next one, test based on drug metabolism. The test for drug metabolism is MEGX test. It is a monoethylglycine oxidilide. And the next one, anti-breath test. And finally, determination of serum enzyme activities. The large number of enzymes estimations are available which are used to ascertain liver functions. They are divided into two groups. One, most commonly and routinely done in laboratory, and second one, not routinely done in laboratory. The first one, the most commonly and routinely done in laboratories. These tests are the two types, and they are serum transaminase is another name is aminotransferase, and the second one serum alkaline phosphatase. These are the tests used for most commonly and routinely done in laboratory, and the second one not routinely done in laboratory. It's the first one serum 5 nucleotidase, and second one serum lactate dehydrogenase, and third one serum isocitrate dehydrogenase, fourth one serum choline esterase, fifth one serum ornithine carbamyl transferase, and the sixth one serum leucine aminopeptidase, and the seventh one serum SHBD, it is a hydroxybutyrate dehydrogenase, eighth one serum aldose and the phosphoexose isomerase, ninth one serum amylase, 10th one serum sorbitol hydrogenase SDH, and finally serum gamma glutamyl transferase. These tests are used in the non-routine laboratory. That's all. That's all. Thank you.